While I'm setting this up, I need to announce that I was really excited to come into this room, or this theater this morning, and see up in bright lights, large-scale inference. I'm not used to having conferences where it's quite that dramatic. And actually, one of the things that helped point out was that, oh yes, not only is it about large-scale inference, but it gave me the subtitle because this is in honor of Brad Thundersoul Efron. So you didn't know that that was his nickname, but it is now. So we can work with that. And I'm going to talk about the type of large-scale inference that I deal with, which is high-throughput biology. Because I'm in a bioinformatics department, as Eleanor mentioned, I've been working with microarrays and things like that for the past, egad, over a decade. And as we've started looking at it, we've said, you know, there's lots of stuff that's really cool here, but in addition to the stuff that's really cool, I want to talk with you today about what I will describe as the soft underbelly of large-scale inference, which is the need for closer and clearer documentation and code to describe exactly what you did. And I'm going to talk about the importance of reproducibility, and this is going to lead to some of the other examples that I have. Now, why is this important, aside from the self-evident thing that it should be there? Well, part of the reason this is important is that I work with clinicians a lot. Several of these clinicians have been studying genes for decades now, and indeed, for some of the genes that they've been studying, like retinoblastoma for cancer, they've got really well-developed intuition about how this gene behaves in a given set of patients. On the other hand, I know that if I give them a list of 100 genes, and these are randomly selected, that I work with a lot of smart people, they'll be able to find a story if I give them 100 genes. And unfortunately, I know this because accidentally we have at least once given people a random list of 100 genes and had them find a story. So that was an accident. But part of the thing there that I mean, the reason I mention this is that this is, to me, an example that because our intuition is poor, then if we are going to use these high-dimensional signatures to guide patient therapy, then we need to be able to trust the mechanisms by which they were assembled. We need to be able to check them, to trace them. And the problem is that if you go to a standard paper in the biological literature and say, wow, that's cool, I'm going to try to reproduce it, you may discover that the methods are just a little bit incomplete. And if you try this, because the problem is actually clinically important, you may be forced into the position of having to employ what we have come to term forensic bioinformatics to say, okay, the raw data is here, the results are here, what must they have done to get from one to the other, regardless of what the methods say they did? So I'm going to try to illustrate that because we've actually found this to be rather informative, and I'm going to try to illustrate this in a clinical example which is of some importance to us at a cancer center, which is, for a given patient, can we predict which chemotherapy is likely to be most useful for treating that patient? As an example, I get a lot of money at MD Anderson from ovarian cancer. That's the main source of my salary there. And one thing we know in ovarian cancer is that most women who come in and present with the disease, they will get the standard frontline therapy, which is a platinum agent plus a taxane. We know, going in, that roughly 70% of the patients we treat this way will see their status improve because the tumor will shrink, they'll feel better for a while, and things like that. We know also that 100 minus that, 30% we are poisoning to no good effect for 6 weeks. Problem is, we don't know which 30% that is going in. If we did, we could give them something else, and we could potentially advance cancer care. And in light of this, there was a paper that appeared at the end of 2006 that got a lot of people really excited because there was a group at Duke that claimed to have figured out precisely how to do this, how to optimize the choice of cancer chemotherapy. Here's how it worked. What they did is they said, we're actually not going to start with patient data. No, actually what we're going to do is we're going to start with cell lines, and not just any cell lines, but the NCI 60 panel of cell lines. The NCI 60 is actually a very special panel of cell lines. It's a panel of cell lines that was assembled by the National Cancer Institute back in the 50s, and one of the things that it's used for is to assess the effectiveness of various chemotherapeutics. So every time a drug company has a new cancer drug that they want to test, it goes off to the NCI and they test it for effectiveness against every one of these cell lines. And they say, okay, we give it to this cell line. Does this drug succeed in killing more of this cell line relative to control than this other drug? And all of this information is publicly available. So what this means is that since this is publicly available for every chemotherapeutic agent we've got, then if we could leverage this to figure out what something says about a patient, then we could potentially expand this to the entire drug repertoire that we've got, which would be really cool. So how'd they do it? Well, they said, we're going to go in, and for a given drug of interest, let's say it's docetaxel, we're going to use this sensitivity information to order the cell lines. There's 60 of them. Actually, there's 59, but that's one issue of data cleaning that we won't deal with. And we're going to say, let's pick out the most sensitive and the most resistant cell lines, say 7 at each end. Then what are we going to do? Well, now we're getting to the large-scale data component. We're going to look at microarray profiles of these cell lines. And again, for the NCI 60, this is publicly available data. And they said, okay, based on those microarray profiles, contrasting the most sensitive with the most resistant, we're going to identify the most different genes using advanced statistical techniques like two-sample t-tests. And they identify the 50 top ones for docetaxel, and they say to these 50 genes, together with their direction of change, that comprises the signature of sensitivity to docetaxel. And now, they say to test this, we're going to go off and we're going to look at microarray profiles of patient samples. And actually, for this one, they used publicly posted data sets. One special thing about this paper, they did no new experiments. It's entirely analysis. So actually, we can access all the data that they used. And they said, okay, for this set of patient samples, what we're going to do is we're going to look at their microarray profile. We're going to see, is the signature there? If we see the signature there, we will say this patient is likely to respond to docetaxel. If we don't see it, the patient's likely to be resistant. That's how it's supposed to work. And the reason this was very splashy, the reason it got a lot of attention, is that they tried it with 7 commonly used agents. They tried it in several data sets, and they reported great results for each. And what this meant for us at MD Anderson is that we got to learn how well-read our colleagues are. Because within 2 weeks, we had lots of people at MD Anderson come to us and say, we would like to treat our patients this way. We'd like to improve things. But there are a few aspects of the paper we don't understand. Can you help us with this? Because they pertain to the analysis. And being nice, helpful bioinformaticians, we said, yes, we will be delighted to. So let's go look at the data and see what is going on. And actually, one of the questions that we started with here was we said, can we verify some of the things that they are reporting? So as an example, we decided to focus on one of the signatures for one of the drugs that they were looking at. And this drug is 5-fluorouracil. And one of the things that's actually in the paper is a heat map showing the signature that they chose based on the cell line data. And that's this heat map that I'm showing you right here. And one of the things that we can see from this is that there's actually a pretty obvious plaid pattern. Each of the 45 genes is either high in group 1 and then low in group 2, or low then high. Now, that's not particularly surprising since we're choosing the genes to achieve this type of separation. These are the ones with big t-tests. Okay, well, we know the cell line, so we said, let's take the cell line data and do our own t-tests. And we did that, and we assembled our heat map, and we said, okay, that looks pretty good. But it turns out there was another thing to go on with the paper, which is the paper also said that these signatures also make sense. And as an example why they make sense, for 5-fluorouracil, here's some of the genes involved. These are known to affect 5-fluorouracil, so on like this. And they gave, as a supplement to the paper, lists of the genes in each signature. So we said, great, let's take the genes they report using for 5-fluorouracil and construct that heat map. And we stared at that, and we said, this makes sense, huh? And we didn't quite know what was going on. So actually, what we did next was we said, all right, these were actually obtained using Affymetrix microarrays. And Affymetrix doesn't necessarily report things in terms of gene names. They report them in terms of probe set IDs, which are mostly numeric. So what we did is we took a list here, and I've listed out the probe sets they report and the ones we found by two-sample t-tests. And as we go from theirs to ours and theirs to ours, is anybody noticing a pattern here? we have an off by one indexing error. And this was somewhat disturbing to us. And actually, what does this say about the intuition about whether things make sense, given that their explanations are in terms of this list, which is unrelated to the biology of interest? Now, of course, this is one drug. They looked at 7. So what can we do? We decided to take a look at the other 6. How are we going to do this? We're going to take the probe set IDs. We're going to move one down. We're going to look at the 2 sample t-test p-values, and we're going to plot them. So let me show you what those are. Well, let's see. Topotecan, they're all off by 1. Etoposide, all off by 1. Adriamycin, mostly off by 1. There's a few we can't explain. Paclitaxel, mostly off by 1. Docetaxel, mostly off by 1. And Cytoxan, we have no idea what they did with this drug. So we looked at this data and we said, this is a problem. How could this happen? So we decided to try to address this, and to address this, we looked not just at the data, we actually tracked down the MATLAB code that they were using. And if you want to read the documentation for that code, you should come to our website, because I wrote it, because there was none in existence before. And in the process of writing the documentation, we noticed something. Specifically, for their software to work correctly, their software needs 2 inputs. The first input is a quantification matrix. These are the gene expression values, which are genes by samples with a header indicating the type of sample we've got. Is it a 0, a training sample that is sensitive, a 1, a training sample that is resistant, or a 2, a test sample whose status is to be predicted? That's input 1. Input 2 is a list of gene names in the same order as the quantification matrix, but, and here's the kicker, for their software to work correctly, this list of gene names must not have a header row. So, if you are working in Excel, and you cut, and you paste, guess what you will get? You will get an off by 1 indexing error. And we tried this with docetaxel, and not only did we get the off by 1 indexing error, we actually were able to perfectly match the heat map, so we're pretty sure that's what was going on. Again, this was run drug. We decided to take a look at some of the others. So let me show you some of the heat maps we can get using their software now. So, topotecan's a perfect match, adriamycin, perfect, etoposide, perfect, 5-FU, perfect, paclitaxel, well, they mislabeled it as cyclophosphamide in the paper, but okay, we see where that's coming from. And cyclophosphamide, we still have no earthly idea what they did with this drug. So that was somewhat disturbing, but at this point we were at least going, oh, wow, we can at least match the heat maps for 6 of the 7 drugs they're looking at. That means we know the genes, we know the cell lines, and this should bother you. It should bother you because while we can perfectly match the heat maps for 6 of the 7 drugs, we only perfectly match the gene lists for 3. The other 3, remember those outlying p-values? They had some genes that we couldn't explain, and it turns out the reason we can't explain them is that their own software isn't producing them. They're coming from somewhere else. So we were disturbed by that. And we said, okay, that's a problem. And actually, we'll come back to that later in the talk. But we got somewhat excited by something else that the software gave us, which is we said, okay, we understand the heat maps, we know what the cell lines are. The software will also give us predictions on a sample-by-sample basis, number between 0 and 1 saying, will the patients respond or not? And that's the real clinical bit of interest. We want to know this. So what are we going to do? We're going to take the software and we're going to try to make predictions. And since they don't tell us on a patient-by-patient basis, this patient got well, this one did not, they just say, here's our overall accuracy rate, we're going to set as a bar that we can use it if we can get predictions at least as good as the ones that they get. So how good are their predictions? So to look at this, we took a look at another plot from the paper, and this is the top panel here, where they're looking at a whole bunch of dots, and I'm going to contend that there's a whole bunch more red dots than blue dots here. And what this says is that we've got a whole bunch of patients who are resistant to adriamycin, and they've got an accuracy of 81% for predicting the correct state. There's just one problem. The problem is that the data that they are looking at came from a paper introduced by Holleman et al. In the New England Journal in 2004. This paper was dealing with the treatment of childhood ALL. This is a type of leukemia for which our cure rates are actually pretty good, near 80%. What that means is that if we have a drug to which most of the patients are resistant, we wouldn't be using it. And indeed, Holleman et al. Thought most of their patients were sensitive. What's going on? Well, they're coding sensitive and resistant as 0 and 1, but they've never written that down explicitly. So in this case, they've gotten them reversed. What does that mean? That means that they would recommend administering adriamycin just to the patients who are most resistant to it. We think this is poor clinical practice. So we are not happy about this. So we also said, okay, this is a problem, this is a problem. Can we get it to work? So we took the samples, we labeled them the way we thought they should be done, and we tried our own principal components plot. And I'm showing you this here, and here we can see one group of cell lines, that's the resistant. Here's the other group of cell lines, that's the sensitive. Here's all the patient samples right here in the center. So when we try it, we can't get it to work. So what happened next? Well, all of this took place over the course of several months, during which we were in constant contact with the investigators, saying, we have this problem, we have this problem, we have this problem. Can we get this fixed? And long about April, we basically sent them a draft saying, we don't think it works. We're writing a letter to Nature Medicine. Can you point out where we've gotten it wrong? And if so, let us know. And in May, they basically said, well, we believe it works, and we wish to draw this to a close. So that ended our interaction at that point. And so there is one follow-up from that, which is that we published this letter in Nature Medicine in November of 2007. And one thing about this being a talk on reproducible research is that you don't have to take my word for it. What that means is that not only can you go and read the paper, but you can download our code, which is in R. You can run our code on the data which we also posted, and you will get the same numbers we got. And if you don't, please let me know, and we'll fix that. Okay? That's a standard. That's one of the things that we think is necessary, because otherwise we're going to run into problems empirically, as we just did. And based on this example, Kevin Coombs and I, who are senior members of the bioinformatics department at MD Anderson, said, guess what? Our department is not a democracy. And we changed the way that we wrote reports. And we said, for the stat analysts, here's how we're going to do it for the things going forward. Now, did this resolve the issue? Well, not quite. You see, in that same issue of Nature Medicine, there was a rebuttal. And the rebuttal said that, unfortunately, the analyses of Baggerly and Coombs are deeply flawed. And that is because we've done our own checking and they just got it wrong. So here are some of the things they got wrong. Our labels for Adriamycin, that's the one with all the red triangles. No, our labels are correct, and we've posted new data on our website to show this. And furthermore, their analyses must be wrong because we've gotten this approach to work again in 2 other subsequent studies, also high profile with editorials, etc. Okay, well, this article, or rather our letter and the rebuttal, appeared on November 6th of 2007. I remember this because I was in a hotel in Bethesda because I was scheduled to give a talk at the NCI the next day about this, and I was somewhat worried that I was going to be talking about something completely wrong. So downloaded some data and said, okay, let's take a look at the Adriamycin data. So downloaded the data that was there. The data that was there for Adriamycin, this is the stuff where they got the labels correct, was actually 144 columns worth of data. 22 columns for their training data, 122 columns for their test data. And actually, we thought about trying to cluster sensitive and resistant, and as a prelude to that, we decided to check pairwise correlations. Pairwise correlations are very useful, aren't they, Catherine? And what I'm showing you here is a plot of where the pairwise correlations are really, really high, 0.99 plus. And down here, for the training data, the cell lines, you see this line of red dots on the main diagonal, and that makes sense. Samples are strongly correlated with themselves. We believe that. Okay. Up here, we see that, but we also see red dots off the main diagonal. What's going on? Well, it turns out that while they report results for 122 test samples, only 84 are distinct, because they've taken some samples and reused them 2, 3, or even 4 times. And right about here, there's one sample that was used 4 times and labeled as resistant 3 times out of 4. So we're pretty sure the labels are not correct yet. So we sent this as a note off to Nature Medicine, and Nature Medicine got back to us and said the authors will post a correction, and therefore we don't need to publish your letter. And indeed, in August 2008, they posted a correction in which they redid the analysis using only the 95 unique samples. The problem is that there were only 84 unique samples to begin with, so of the 95 that they then corrected it to, only 80 are distinct, 15 are present twice. Of the 15 present twice, 6 are labeled both ways. So the correction of a correction still needs correcting. So we decided then that, okay, that's a problem, but let's move on to the other validations. So in particular, one of the other validations was a paper that showed up in the Journal of Clinical Oncology, and here they decided to expand the approach to deal with 2 new drugs, namely cisplatin and pemetrexed, both of which are used in treating lung cancer. And they measured these things and the cell lines using AFI metrics, U133A arrays, and one thing that made the story very plausible here is that they said that the gene lists that they identified for cisplatin included ERCC1, ERCC4, and DNA repair genes, and there had been a recent set of papers in the New England Journal of Medicine talking about how these genes are associated with how cisplatin is processed by the body. So this made things look really plausible. So we looked at that. We looked at the data, and with some work, we were able to perfectly match the heat maps. That means that we know the genes, we know the cell lines. And it turns out that for cisplatin, we can match 41 of the 45 genes they report, after we account for the off by 1 error that's still there. There's 4 that we can't match. Do I have any guesses from the audience as to the identities of those 4? The 4 probe sets that we cannot match are ERCC1, ERCC4, a second probe set for ERCC1, and a probe set for FANCM, which is a DNA repair gene. Now, even in this context, it turns out that these last 2 probe sets are special. What makes these 2 probe sets special? Those 2 probe sets are special because they are physically not present on the arrays that were used. 2 of their best probe sets have never been measured. We thought this was disturbing. We wrote a letter about this to the Journal of Clinical Oncology. JCO said, "We're sorry, we can't publish this." We sent a second letter saying, "Could you tell us why?" They sent a second reply saying, "We're sorry, we can't publish this." So we moved on. We moved on next to their next validation paper, and this is a paper by Bonnefoy et al., where they said what the extension is here is now we're going to treat not just one drug, but we're going to deal with combination chemotherapy. And this is important because most patients don't just get one drug, they get several. And in particular, they claim that the improvements that we would have seen had we been using their approach would have driven the response rates up from 44% to 70%. That's a big deal. So we looked at the data, and actually we downloaded it from Geo. And actually, before we got into any real deep analysis, we also said, by the way, is there any reason that these groups might be different aside from the biology? So we decided to take a look at pairwise correlations again, and I'm going to show you I've set the cutoff a bit lower here, but I'm going to contend that there's roughly 3 clusters in this set of data. And it turns out that we had an idea what these clusters might be. You see, these were Affymetrix cell files. We took the cell files and we processed them to extract not the numbers, not the quantifications, but the header strings which contain the date on which these arrays were run. So over here, I'm plotting these things as a function of run date. Here's cluster 1. The black X's there are half of the patients treated with FEC. Greens were excluded from the analysis. Here's the other half of the patients treated with FEC on day 2. Here's all the patients treated with TET, where all of these samples were run on a different machine, in a different lab, in a different country. There is complete confounding of the experimental design with the biology of interest. There might be some differences there. We wrote this up together with some other problems and sent this off to the Lancet Oncology. The Lancet Oncology responded that we appeared to be arguing about a statistical issue to which there is no right or wrong answer, and as a result, they did not see the need to publish our letter. So we said, okay, we will move on. And indeed, in 2009, some friends of ours who were working in melanoma said they've now extended this to work with temozolomide. And they showed us the heat map and said, can you help us here? So we said, okay, we're good at matching heat maps. So we tried, and we failed. And that was very distressing to us. And it was so distressing that we decided to just go back and do something odd, which is look at the literature. And when we looked at the literature, we also noticed the heat map they'd published for cisplatin 2 years before. And all of a sudden, we weren't too bothered by the fact that we couldn't match this one anymore. Turns out this paper achieved a neat hat trick. This paper was nominally about temozolomide, drug A. It included a heat map for cisplatin, drug B, and included a gene list for an entirely different drug we still haven't identified. We reported this to the clinical cancer research, and a new figure was supplied which still doesn't match the gene list. Now, all of this went through 2009. This is going on 2-3 years now, and this is where the story gets bad. The story gets bad because in June of 2009, is when we learned that they'd actually already started clinical trials in which they were using this approach to allocate patients to treatment arms. In some of these clinical trials, the drug signatures they were using were ones where, based on the data they'd posted, they had the sensitive and resistant labels reversed. We were not happy. We compiled all of our results, wrote them up as a single paper. We initially tried to get to the doctors again, so we circulated a draft to the editor of a biological journal, who got back to us and unfortunately said, well, the story's a bit too negative. Can you fix that? And we said, no, we don't think we can. So actually we did something new, which is we said, well, we're going to send it to a statistics journal, and we sent it off on the 1st of September to the Annals of Applied Statistics. And on the next day, I got email from Brad Efron. saying that he was going to handle the review of this paper himself, and I'm going to frankly admit right now, I took that as a good sign for the paper. And indeed, 2 weeks later, we were informed that the paper was accepted as is and was published online at that time, which means that people should send more papers there, because two-week turnaround is pretty good. And so we were really excited by all this. And this is when things began to happen, because in addition to coming out in the journal, once it was out in press, we also mentioned it to certain reporters. And at the start of October, it was covered by the Cancer Letter, which is a community journal for the cancer community. And it turned out that at the start of October, Duke announced that it was going to run an internal investigation and that it was suspending the clinical trials pending that review. And we breathed a sigh of relief. The next public news came in January of 2010. And that was when Duke announced that it was getting ready to restart the clinical trials. That was because they had finished their investigation, and on the basis of their investigation, they had strengthened their confidence in this evolving approach to personalized cancer treatment. We were surprised. We were surprised in particular because the data that was still out there was still wrong. So we asked, could we see the data that you showed to the external statistical reviewers that you commissioned, because they must have seen raw data to get it right, or could we at least see the report justifying the restart? And Duke said no. They said, while the reviewers approved of our sharing the report with the NCI, we consider it a confidential document, and a future paper will explain the methods we are using. This is for methods which you're using in clinical trials and have been for 2 years by this point. So we weren't happy. And as it turns out, there was another thing, which is that when Duke announced that they were restarting the clinical trials, there was something else there that they didn't mention. So what didn't they mention? Turns out that the trials were suspended in October and restarted in January. In November, while the investigation was underway, the lead investigators, Drs. Potty and Nevins, posted brand new data to the web for cisplatin and pemetrexed, two of the drugs that we were worried about. So we thought, oh, well, maybe they've cleaned it up. So we downloaded it. And it turns out that for cisplatin in particular, they posted 59 samples, microarray profiles of them, where these 59 samples were the ones that they had used to validate the performance of their signature. And these 59 samples came from a larger data set available at the Gene Expression Omnibus. This is a public data set with 153 samples in it. So we said, okay, let's match the ones they've got to that larger data set. And we're going to use pairwise correlation again. So if we've lined everything up correctly, we should see 59 red squares all on top of that blue line there. So how many red squares are on top of this blue line? 0. As it turns out, there's only 43 red squares there at all. 43 of those, what that means is that for 43 of the samples, we can tell you which array they're getting the numbers from. But they got the name wrong. And they also got the clinical information wrong. For the other 16, you don't see a red dot there. Why is that? That's because for those 16 samples, they've scrambled the gene names so badly that we have no idea what the samples are. But what I can tell you is that all 59 validation samples are wrong 2 years after they started running clinical trials using this drug. We reported this to Duke in November. We also reported this to the NCI the same day. And we said, do you have any comment? And Duke had no comment. So there was the question of where we could go next. And then we said, well, wait a minute. Duke wouldn't show us the data. But can we get it? See, when they told us they wouldn't give it to us, they said something interesting, which is that while the reviewers approved of our sharing the report with the NCI, I want to pause here. The NCI is a federal agency. What that means is that in this country, it is subject to the provisions of the Freedom of Information Act. So, in April, we applied for, or Paul Goldberg of the Cancer Letter applied for this report under the Freedom of Information Act. It was supplied in May. We looked at it and we said, guess what? This report justifying the restarts makes no mention of the fact that all the validation data were wrong. We think that that probably would have merited a mention. What the heck's going on? Duke had no comment. We asked the NCI if they had a comment, and they told us that, oh, well, based on concerns that have been raised, we have started reviewing signatures in use by this group, and while we cannot comment on the clinical trials you are discussing, we are in the midst of pulling a signature from a phase 3 clinical trial from a different group, or from a different study, because we no longer trust its validity. So this is one way of the NCI saying we're not happy. So what would happen next? Turns out what happened next, and this is a catalyzing event, is not necessarily scientific in nature. Happened on July the 16th of 2010. And that's when the Cancer Letter reported that Anil Pati, one of the PIs, used to claim that he was a Rhodes Scholar on grant applications and various other things. The Rhodes Foundation disagreed. This was disturbing. And he was placed on administrative leave that day. Now, there had been a number of statisticians following this because I've been given this talk for years, and one or two of them had been talking about writing a letter. So the evening of the 16th, I sent email to Rafael Irizarry, and I said, you know, if you're going to write a letter, now's about the best time you're ever going to have. So that weekend, he and a bunch of others put together a letter that went off on Monday the 19th and landed on the desk of Harold Varmus, the brand new head of the NCI, saying, would you please suspend these trials until we can figure out what the heck's going on. As a side note, I will note that one of the signers of that letter was Brad Efron. Now, what happened next? Well, Varmus and Duke got together and they said, we need another investigation. We're going to ask the Institute of Medicine to take a look at this because we want to have it outside of Duke. And a month or two later, the Institute of Medicine said yes. Then things progressed. Things were quiet for a while. And on October, there was the announcement that the Duke investigators were initiating calls to retract one of the papers. Which one? The one dealing with cisplatin and pemetrexed. Why were they retracting it? Because on closer examination of the validation data, it turns out that 43 of their validation samples were mislabeled and they could not match the other 16 to their database. We'd told them this the previous November, but then they restarted trials. In November the 9th, Duke officially terminated the trials and announced that in retrospect, these trials should never have been run. And in mid-November, they began retracting other papers and Anil Pati resigned from Duke. What's happened since then? Well, the Institute of Medicine began its reviews, and it began on the 20th of December. And in my experience, first meetings of committees tend to be boring. This one wasn't. It wasn't because at the December 20th meeting, Lisa McShane of the NCI Biometric Branch got up and said, let me tell you about the NCI's interactions with this group for the past 4 years. By the way, I'm going to back up my presentation with roughly 550 pages of documentation, which the NCI is now releasing publicly. There was a reporter there at this meeting. He sent me all 550 pages 2 days later with a note saying, Merry Christmas. So I have now read all 550 pages, and some of the interesting things there is that the NCI was quite upset, and it was, amongst other things, upset because in April of 2010, it learned for the first time that it was actually providing funding for one of the clinical trials in question. The only reason it hadn't acted before is that the NCI doesn't have regulatory authority for trials it does not fund. It didn't know this because clinicaltrials.gov did not list NCI support, which is the responsibility of the institution. They found out that they were. They said, we would like to see the data and code, please. At the end of June, they invited Duke investigators out to the NCI and they said, this does not support running the clinical trial. By our reading of the documents, the NCI was within a few weeks of unilaterally shutting down some of these trials over Duke's restart objections. So, yes, without the Rhodes Scholar story, things still would have stopped. But the Rhodes Scholar story came out 2 weeks later, so it didn't have to. So, beyond that, the IOM has been continuing to review. And if you really want to see something about how biomarker studies are being reviewed right now, I recommend listening to some of this. There's MP3 audio from all these meetings. You can go to our website. Slides are available. This is a clickable link. There's roughly, 20-some hours of listenable audio here, and some of it's rather interesting. And on August the 22nd, they had a presentation from Duke where they said, how bad is this? Well, they've now surveyed all 40 papers, research papers with Pati's name on them from Duke. In their estimation, roughly a third will be retracted in full, another third will be retracted in part, and the remaining third they think are going to be okay. 7 have been retracted to date. We also found out what happened with that report where we talked about the validation and that not working. Turns out that we sent it to the deans at Duke overseeing the investigation. They forwarded it to the head of Duke's Institutional Review Board, who's in charge of overseeing patient safety. He forwarded it to Drs. Pati and Nevins, saying, is there anything new here? Dr. Nevins argued that showing this report to the external reviewers would bias the review. This argument was agreed to by the Duke deans, the head of the Duke IRB, and the head of the Duke medical system. So they restarted trials on the basis of a report where the experts writing the report were never told that all the data that they were looking at was mislabeled. That's a problem. Now, the story is not over yet, and it's gotten just a little bit of play. So what this means is that for now we have a bit of a soapbox. Now, this is not an unmixed blessing, because some of the stories are saying, my gosh, genomics is crap. And that's not the message. The message is that we need to be careful about doing this because it's very easy to make simple mistakes. But we can do it right. So how can we learn from this? How can we move things forward? Well, I want to give you some observations and lessons here. First off, this case that I have just discussed is pathological. We do not see cases with this many mistakes on a regular basis. No. But the component mistakes, several of those we've seen before. We've seen them at MD Anderson. You will never see them because we saw them first and they didn't get out of the institution. But what are these most common mistakes? The most common mistakes are simple ones. Ones like complete confounding of the design, or mixing up row labels, or mixing up column labels. These are easy mistakes to make in Excel. And if the documentation is clear and complete, then actually finding simple mistakes is easy and correcting them is also easy. But if the documentation is incomplete and it makes it to the literature, then uncovering the errors becomes hard and requires some of the reverse engineering we have just discussed. So what do we think should be required in terms of reproducibility? Well, in our group, we have been writing reports in SWE, which is a combination of R and LaTeX, since 2007. I'm actually going to assert that the reports that we've written in that period are now completely reproducible. We can run the code. We can still get the same results. I will note, I am not contending that every report we have written in that time is correct, just that we can check it. Now, for papers, I got together with various folks, about 40 or 50 of them actually, and we wrote a note to Nature saying, here are some things we would strongly recommend. And since Nature doesn't allow 50 authors on some of these things, it means that my CV says Baggerly, so okay. And things we look for are the raw data, which we've discussed, provenance. What are the labels? Where did you get them? This also includes for us the clinical data. There are some cases of array data out there where we have the arrays and we don't know which ones are sensitive or resistant. Those data sets are less than useful. At this point, we think we also need code. Verbal descriptions are no longer adequate once the dimension gets this high. We'd also love some of these others, but we're not necessarily as willing to fight for these. Now, these are recommendations we're going to make for papers. For clinical trials, we're willing to be a bit more hard-assed. These are not recommendations. We think these should be requirements. Now, in the end, I'm actually somewhat optimistic. Why? What are some reasons for hope? Well, number one for us is our own experience. We've been doing this for years. It is now automatic in our department to make things pretty reproducible. And actually, it's not taking that much extra time. The biggest change was shifting the mindset to getting used to writing things this way. It took us about 2 months to make that shift. Second reason is that actually Duke's now taking this seriously. They're trying to clean it up. And they have a translational medicine quality framework team, which is putting together some pretty darn good guidelines. And they've given some useful talks, and we're very happy to see this. Things were screwed up before, but they're honestly trying to make it better now. The NCI, earlier this year, held a workshop on what should be the guidelines for the use of complex signatures in clinical trials that it funds, where it sets the rules. Next week, in Brussels, the NCI will be presenting for the very first time some of the recommendations from that workshop. These include, oddly enough, that if you have a complex signature and you want the NCI to use it, that, guess what? Somebody in your group should be able to supply the NCI with the code and the data from which it was derived so that they can run it. The Institute of Medicine is writing its recommendations now. Those will come out in early 2012. And some of that we'll be talking about working with the FDA to make new requirements for complex signatures involving IDEs. IDE is short for Investigational Device Exemption. According to the FDA, a microarray signature, a complex mathematical assemblage, is a medical device. It guides treatment in a risky setting. So what this means is that by imposing a requirement for IDEs, they can get essentially the same type of information the NCI is asking for. So we're actually pleased to see that. And one other reason we're pleased to see that is with the IOM and the NCI coming out about this, that letter that I wrote before showing recommendations, that was mostly statisticians. We were happy to have that support, but it wasn't enough. To get this moving forward, we want to have support from the broader biological and other communities, and that's why we're happy to see movement from the NCI and the IOM. Finally, there are journals. Can we get journals to make things more reproducible? Can we get them to ask for code? Can we get them to ask for data to be posted? We're optimistic there because, well, somewhat as a result of this example, we've been going around the country and we've been speaking to a number of journal editors, talking about what things would help make these more reproducible. And it also helps to be able to point out the fact that there were some cases where they didn't lessen and trials kept going. So what are we recommending? At a minimum, we'd really love to see them asking for the data and code, and we're not necessarily requiring that they host the data and code. Links would be fine, but we will ask that after acceptance and before publication, somebody at the journal actually clicks on those links and says, oh, there is data here, and oh, there is code here. Not reviewing it, just checking that it exists. That, we think, would move things forward substantially. Those are my reasons for hope. That's why I'm optimistic. Now, I have to finish up here, so I need to acknowledge the fact that I didn't do all this myself. This actually took a lot of time. Kevin and I estimated that between us, we sunk about 2,000 man-hours in reverse engineering all this. It shouldn't take that long. We also note that we had a lot of folks funding this. This was important. This is because when people come to us and say, we have questions about a paper, normally we'll spend a few hours on it and then we'll stop. Here, PIs of several large grants said, we wanted to know how this works. That meant we said, okay, we'll charge some of our time to this. And that's how it got to happen. Finally, I'm going to say that if you want to learn more, you should read this Annals of Applied Statistics paper, which, in my own opinion, is pretty good. And this will be good because you can read it, you can cite it. That will make me famous and my mom happy, and that's a good thing. And you can also learn more about reproducibility. And if you want to read more about updates, there is a link to our website, and that's because some of the developments since our paper have not actually been in the standard statistical literature, so I'm just pointing you to there. And I have one last acknowledgement, which is to thank you, the audience, for putting up with me for the past 40 minutes. Thank you very much. Okay.